Dear students, each short lecture helps to you to understand some test control exam questions and help to provide to training exam. I represent to you 12 examples which help to you to provide training before exam, and I hope help to you to preparing to exam. Example number 1. On its example, ask about spherical vesicles in cell inside in cytoplasm, which surround by membrane and include hydrolytic enzymes. We have some questions about its organelles. It's a single membrane surround organelle, include hydrolytic enzymes which release in digestion process inside in cell. And sometimes its question include about which is organelle to provide digestion, or which is this organelle surround one membrane and include hydrolytic enzymes. Always in its answer must be present lysosomes. Lysosomes just one organelle inside in cell which provide digestion process. Example 2. Questions about special organelles which provide protein synthesis. It may be questions about subunits in cytoplasm, two subunits organelles, or maybe questions about formation of its subunits via in nucleolus, or maybe questions about nucleolus organizer present in chromosomes 13, 14, 15, 21, and 22, and it's special regions in the chromosomes which provide synthesis are RNA, ribosomal RNA. And in its cases, always you can write about protein synthesis in ribosomes, or maybe which is organelle produce protein is ribosome, or maybe which is place formation of ribosomes is nucleolus. Example number 3. Example show to you test ask about phases in mitotic division. Maybe questions about cell cycle. It's questions ask, for example, which is a period when we have replication of DNA. It's S phase in cell cycle. Or maybe which is period which release more tubulin proteins. It's a post-synthesis phase or G2 phase in cell cycle. More questions about phases in mitotic division. Location chromosomes in equatorial plate is metaphase. Maximum condensative chromosome in metaphase, and always in karyogram use chromosomes in metaphase period of mitotic division. Maybe questions about anaphase. In anaphase, chromosomes move to poles and form specific elements with centrioles and mitotic spindle. In anaphase, always separate chromatids to peripheral poles. And maybe questions about telophase, late and early telophase. In late telophase, finishing process of cytokinesis. Example number 4. Its example show question about organelles with two membranes which provide ATP synthesis and ATP synthetase enzymes, and or maybe questions about process formation of ATP, or maybe questions about which is organelle include DNA and specific ribosome. It's all questions told and asked about mitochondria. Just one organelle in human cell include two membranes, is mitochondria. Example number 5. It's question about recessive gene and if we have in family recessive gene in heterozygotes or maybe if we have parent not carrier of gene with recessive gene will be represent in future generation or not. Students, it's questions told about first rule of Mendel. When we have a recessive gene may be represented in 25% in future generation if it's heterozygote parents. Or maybe if we have homozygote parent dominant and heterozygote parent second, not representative recessive gene. Another question, if we have a question about specific dominant genes. In dominant genes, just only in 25% recessive alleles not representing some diseases. Maybe questions about chromosomal destruction and questions about changes amount of chromosome. I repeat to you and help to you. You see, if we have just one chromosome from pair is monosomy, or if present plus one chromosome, it's trisomy. And in normal, always two chromosome in one case, and in general, 46 chromosome. Example number 6 asks about representative recessive gene and why maybe represent in future generation gene. Just if we have heterozygotes parents and its recessive gene not visible in heterozygote but maybe form in future generation. Example number 7. Its examples ask about changes amount of chromosome and for example show when we have 22 plus X chromosome, in other words it's monosomy by X chromosome. Here it's a Turner syndrome. Correct answer because just in Turner syndrome present X0 position of chromosomes in organism. May be present some questions about amount of sex chromosomes. Klinefelter disease when we have XXY and if we have trisomy of X chromosome will be in questions about XXX or maybe a question about Klinefelter syndrome I told XXY or maybe question about Edwards syndrome XYY. Example number 8. Here are asked about embryonic development and position of step-by-step stages of embryogenesis. Here you see the first fertilization, cleavage, next gastrulation, and after gastrulation before organogenesis and histogenesis neurulation. Neurulation is process formation of neural tube and always after gastrulation. Maybe questions about germ layers and origin of some organs, tissues from germ layers. Please, in its case, see in information in textbook of histology, cytology, and embryology, and you can see in specific new textbook histology, cytology, embryology must have to examine. I told on group about its new book, Help to You. Example number 9. Its example question show and ask information about types of ovum. Please look very attentive. Here you see types of ova and ask about its types. Which is type ova when we have more yolk or not more yolk? Some questions about types in human. In human we have secondary oligolycetal, isolycetal, ova. Telolycetal ova include more yolk in vegetative pole. Example number 10. It's example ask about complementary triplet and it's a question show answers about complementary pairs. You know G and C complementary between 1 by 1 and A complementary to T in DNA, but A complementary to uracil in RNA. Its question asks about tRNA. Here you see correct answer. Example number 11. Its example asks about main enzymes which include in process of replication, maybe questions and about enzymes in process of translation, and use place your knowledge about types of enzymes. Here ask about process of replication and formation of two individual strands. Helicase main enzyme which will be separate chains one by one and to provide and help in process of replication. Example number 12. Its question asks about process formation of messenger RNA, what's happening in its process, and please look very attentive and recognize correct answer to processes formation of messenger RNA and processing. It's a process formation of messenger RNA from precursor of messenger RNA. In its case, maybe ask about intron, exon, and processing process, which is function of intron in some questions, function of exon, and what does it mean translation? Process formation of protein in cytoplasm together with ribosomes, together with messenger RNA and transport RNA. Thank you for your attention, my dear students. I hope its video helped to you to preparing to exam.